You know what metabolism is, the two main branches. And specifically, in the muscle, when we consider energy metabolism, these are the processes that are involved. These are the things that make ATP, that sustain contraction. Creatine phosphate, or phosphocreatine, however you want to say it, glycolysis, and then aerobic metabolism, or respiration. And as I lay these out for you, we'll talk about each in not a lot of detail because it's the focus of ex-phys and biochemistry and metabolism. This is a primer. We're going to hit the highlights for each of these three, but I want you to understand how they work and how they are orchestrated together. And this is supposed to, there we go, look like so. What I want to show you here is how they respond to exercise. If you, sitting at rest, decided to get up out of your seat and go for a bike ride or go for a run, and you're just jogging for an hour, how would you be able to accomplish that? How would you not run out of energy? How would you replenish the ATP stores? And this is what the figure here is going to show you. So the light shaded white box in the background represents that you need to do the work. You get up from your seat and you start running and you're at a certain workload. Sitting here right now, you're at a certain workload, but it's really low. You need a certain amount of energy. If you started sprinting, you need a much higher amount of energy. And that doesn't change. If you are maintaining a given workload, the demand for energy is constant. So how do we meet that demand is what I want to show you here. This is a shock. To you, sitting here at rest, to start exercising is a shock. Nothing starts at the drop of a hat. So the first thing that you call on is free ATP in the cell. You have some in the muscle. It's available for use, but you don't have very much. 3 to 5 seconds before you run out. But it can do the job of giving you energy in the meantime while something else takes over. So think about what this is showing. It's not showing you that ATP runs out. This is showing you that initially ATP in the muscle provides all of the energy to do the work. And then very quickly, something else takes over and it provides no energy. It doesn't run out. In fact, it hardly ever changes. It just doesn't supply the energy after a few seconds. Other things have taken over. But initially, the first shock as you transition to exercise relies or requires you to call on ATP in the muscle. What is the thing that takes over really quickly? Phosphocreatine. You have ATP in the muscle, you have phosphocreatine in the muscle, and that system very quickly turns on to make new ATP. This is largely what takes over for the free ATP in the cell. And so very quickly after you jump out of your seats and start to jog, a large percentage of that exercise is being supported by phosphocreatine. But you also don't have very much phosphocreatine, and other things need to take over. Not long after, 30 seconds later, something else is taking the brunt of the energy provision role, and that thing is glycolysis. Glycolysis takes a little bit longer to turn on. There are many more steps. Phosphocreatine has one step. There are many more steps, and so it takes a little while, but it can provide a lot of energy. And it does that. In the first 30 seconds to a minute, the majority of energy is being provided by glycolysis. Now, importantly, even though the systems are changing, you're getting energy from different systems. At any single point in time, they will all add up to 100%. That's the demand. So for instance, take this slice of the graph. Early on, when you're relying mostly on phosphocreatine, there's also a small reliance on some ATP and a small reliance on glycolysis. Overall, you add all of those contributions up and you satisfy the workload. It equals 100%. The exercise requires energy at a certain level and your muscle responds accordingly. All of these so far are fairly short-lived. If you wanted to run for longer than a few minutes, you need a system that can provide energy long-term, and that is aerobic metabolism. After a couple minutes, and that reminds me, I think if you are looking at the slides, the scale at the bottom is slightly different. Make sure that you note the difference in scale. This is seconds and not minutes. That was a typo. And then these numbers have changed. I uploaded that before I had finished configuring this graph. After a few minutes, if you're jogging, your aerobic metabolism can satisfy everything. You don't need contributions from the other systems. If it's a moderate endurance type workload. It's different if you're sprinting. It's different if it's weightlifting. This is jogging, going for a casual bike ride, exercise. Yeah, question?
The scale on the bottom is minutes?
The scale is in seconds. It's written in minutes, I think, on the slides.
Oh, okay.
If it's not, great. I don't want to confuse you. It's supposed to be in seconds. So this is 1 minute. This is 2 minutes, 3 minutes, and then so on. What I want to get across with this graph is that they all turn on together. You can't selectively activate phosphocreatine. You can't selectively turn on glycolysis and nothing else. All of these systems turn on together. They have different, what I'll call lag times. It takes each of them a different amount of time to get fully operational. But they all turn on together. It's not selective. You can't pick and choose which one you turn on. And they don't run out. You don't switch from one to the other. Phosphocreatine doesn't run out and then you switch to glycolysis. Glycolysis doesn't run out and then you switch to aerobic metabolism. It doesn't work that way. They all turn on together. You can emphasize one or the other, but you'll learn about that in XPhys. I just want to get that point across now. So let's take a little look at each of these main systems. Not ATP, because we know what that looks like. We saw that when we saw the muscle contracting. Phosphate broke off, we got energy. Easy. Creatine phosphate is also pretty easy. One more step is involved here. Creatine phosphate is a supply. It's a reservoir for phosphate. And if you're sitting at rest, you don't have a very high energy demand, there are enzymes that will stock up that reservoir. And the enzyme in question here is creatine kinase. It's not shown on this figure, I'm realizing now, but it makes this happen. It takes any creatine in the muscle. If ATP is present, it will transfer a phosphate group. That's what the enzyme does. So it stocks up the reservoir of creatine phosphate in the muscle. If you're sitting at rest, you should be fully stocked with creatine phosphate. And this is always happening to some degree, but there's not going to be a lot of energy demand at rest. If you're sitting there, you don't have a very high need to contract your muscles and use energy. And so the energy that your body stores or your muscles are normally producing is used to make creatine phosphate. And at some point, it will slow to a trickle. So you might just have a reservoir in the muscle. This is the reservoir that you call upon in those initial few seconds. What does that look like? Well, you jump out of your seats, you start jogging down the hallway, muscle starts contracting, which we know means ATP starts getting broken down. Where does that ATP come from? If ATP is broken down, it essentially pulls this reaction upwards on the slide. It's being broken down over here to sustain contraction, and now we need to resupply, replenish, restore the levels of ATP that were there originally. So ADP combines with phosphocreatine. That phosphate group transfers back, and it acts as a reservoir. This very, very quickly will replace the ATP that the muscles use. There's almost no change in the concentration of ATP in the muscle because this is so quick. Instantaneous. As soon as you start breaking down ATP, this reaction occurs. Creatine phosphate donates a phosphate group. But because you only have so much creatine in the muscle, you don't have a large reservoir. It happens very quickly. It allows you to transition, but it can't sustain exercise for very long, so something else has to take over. You will potentially run out of creatine phosphate if you exercise hard enough. And yes, this is the same creatine you've heard about in supplementation spheres, or in weightlifting spheres. Different role there, and we can talk about that later. It's not the point of the lecture. Provides a reservoir for phosphate that allows you to make ATP quickly. Before this runs out, we turn to our second major, or we involve our second major energy provision pathway, which is glycolysis. Now, I've edited this slide to reflect what I think is a more accurate depiction of what actually occurs. Creatine phosphate doesn't run out. Maybe if you're sprinting and you do repeated sprints, you'll learn about a Wingate test when you do XPhys. Maybe you run out then. Hardly ever will you experience depletion of creatine phosphate. Because everything turns on at once, you don't have to wait for creatine phosphate to deplete. You simply start to use glucose. You can bring in glucose from the blood glucose, or you have some stored in the muscle as a molecule called glycogen. Both of those things will supply glucose, and glucose is converted in glycolysis to pyruvate to generate ATP. It's not converted to pyruvic acid, and it's not converted to lactic acid. So I'm going to edit this diagram too. I would cross off this thing at the bottom. You don't want lactic acid there. You don't want lactate and into the blood. I'll come back to that. And you create two pyruvates, not pyruvic acids. Those don't exist in the neutral pH of your body. And it doesn't matter. Our interpretation is intact because this is where ATP is produced. It doesn't matter what happens down here. Glycolysis makes ATP. And this would go to sustaining muscular contraction. It provides ATP just like creatine phosphate. Now, glycolysis is always active when you're moving around, when you're exercising, just like creatine phosphate would. It turns on at the same time. Glycolysis is always active. And what's important to understand here is that it has a very high potential for flow, for flux. This pathway can move really quickly. Whatever activity you have in glycolysis sitting there, it can be 300 to 400 times faster when you're exercising. Incredible potential in this pathway. And so even though you only get 2 ATP every time you go through this pathway, you can renew that and do that many times, very quickly, over and over again. Lots of ATP or energy generation potential. A note about anaerobic glycolysis. Anaerobic means what? No oxygen. Anaerobic means no oxygen. Yes. I want to give some context for why anaerobic glycolysis is named the way it is. The processes here don't use oxygen, hence the name. Anaerobic glycolysis does not involve oxygen. That is fair. That's fine. The problem sometimes is that people interpret anaerobic glycolysis as the muscle not having oxygen, which is an important distinction and is never the case. There is always oxygen available in the muscle. The muscle is not anaerobic. The pathway is. So glycolysis is always active, and then pyruvate can do some things. The next thing that we're going to see is one of the endpoints of glycolysis. These pyruvates that you make can go into the mitochondria. And that's the next major energy pathway that we're looking at. So I'm showing you that here to emphasize that connection. It is also possible for pyruvate to make lactate. And it all depends on the flow rate. I think about it as a sink. You have a sink where you do your dishes normally. It's got a drain and it's got a faucet. And you turn the faucet on a little bit and there's water that flows into the sink. Right? You turn it on a little bit and the water's going to drain right out. No problem. You turn it on more, higher flow rate, the sink starts to fill. If you have a magic faucet where you can turn it up indefinitely, big time filling, and some spills over. Lactate is that spillover. Pyruvate always wants to go into the mitochondria. The mitochondria is the drain in this analogy. It's just really slow at doing that. It's the most efficient way to make ATP, and it's trying. It turns on with creatine phosphate, with glycolysis. It's just slower because it's a more complex pathway. Pyruvate wants to go into the mitochondria, but if this flow rate is way too high, some spill over. And that spillover is what produces lactate. I'm not saying lactate doesn't exist. You will obviously produce lactate if you're sprinting or working at a really high intensity. Stop and go sports. It's all based on the matching of these two pathways. Now I'll come back to lactic acid in a second, but I want to go first to respiration. If everything works out and you're going for a jog, did you not get that? I see some confused faces. Okay. I'll go back. Flow rate's too high, some spills over, lactate's produced. If you're going for a jog and everything works out, we want all of this pyruvate to go into the mitochondria. That's where we get the most bang for our buck. The highest energy yield per molecule for whatever you're inputting into the equation. Cellular respiration is what is responsible for that very efficient generation of energy. And it's not the focus of this class to look at the enzymes in the electron transport chain or to look at the TCA cycle. You're going to have a lot of exposure to that in classes coming up. But what I think is important is that first we acknowledge really any food stuff. Your food gets broken down and the terminal endpoints all go into the mitochondria to be processed. Pyruvate's one of those. And it's the job of the mitochondria to harvest that food. And oddly enough, what they're harvesting are electrons. It sounds made up. It sounds too high tech, like a computer. But your mitochondria are harvesting electrons. The electron transport chain, which is what the ETC is harvesting the energy from the electrons in the food that you eat. So pyruvate goes in, question mark happens, energy comes out. We're leaving it at question mark in this class. One way that I like, that I think really exemplifies the flow or how the mitochondria works is water flowing downhill or a ball bouncing downstairs. Essentially what happens is electrons get progressively transported into lower energy states and eventually they form water and they can't be reversed at that point. It's irreversible. You form water and it's a latch. Very efficient, progressive extraction of energy from this system, but the details of which will remain a mystery to you until you get to a more advanced course. Something happens in the mitochondria. We make a lot of ATP using oxygen. So food electrons harvested for energy, they're ultimately transferred to water. That can't be reversed. The end for now. This is by far the most efficient means of making ATP. A brief note now on lactic acid. Lactic acid. Because inevitably there are questions about it, or you hear it spouted off in general media, or massaging gets the lactic acid out of your muscles. That's a crock of crap. Lactic acid doesn't exist in the body. What is an acid? Acid is a substance that can donate a proton, that can donate a hydrogen ion. And so for something to be acidic, it just has a high concentration of protons. That's the definition of acidity. So an acid is a substance that donates protons. If something is acidic, it has a lot of protons floating around. Pyruvate and lactate don't do this. There is such thing as pyruvic acid and lactic acid, but they don't exist at the neutral pH of your body. You're very neutral. You're in the middle of the range, right about 7. These things are not acids at a pH of 7. You'll be an acid at a pH of 3, but you would be dead long before that. These are not acids in our body. And in fact, the spillover forming lactate actually removes protons. And I'm going to show you that quickly. I know it's not biochemistry, but I find it fascinating. Any spillover actually takes protons out of the muscle. Well, hold on. If an acid donates protons, and if something is acidic when there are a lot of protons hanging out in solution, if I take protons away, it becomes less acidic. Lower proton concentration, less acidic environment, which is a higher pH. Making lactate actually protects against acidity. And I just want to show you where that comes into play. This is the reaction, and I don't expect you to copy it all down, but just follow the colors. This is what happens at the end of glycolysis. This is the thing that goes into the mitochondria. And if there's spillover, this happens. This is the spillover reaction. Here's one reddish pink H, and there's another one. And this is importantly where the acidity comes into play. This is normally floating around, very acidic, consumed by lactate. It's not floating around. Therefore, the surroundings are not acidic. So the big question is, what causes the acidity that you so commonly think of as lactic acid? That's the million-dollar question. Luckily, we get to that as we discuss fatigue. Now I'm hearing a lot of murmurs and rumbles. Is this just upending your world? Well, my grade 11 teacher told me lactic acid was a thing. Did they said that? Yeah? That's wrong. Lactic acid is not a thing. Where does the acidity come from? We're going to find out. Because acidity is one of the major reasons that muscles fatigue. That you can't contract forcefully, repeatedly, in very quick succession. Acidity turns off enzymes within the muscle. It's bad. We'll talk about where that comes from shortly. So fatigue as a concept might be what you're feeling now. End of the semester, I'm throwing new slides up that you don't have in the slide set online. Fatigue. From a muscular point of view, the same activation doesn't yield the same force. Or you can't activate the muscle as much as you could before. That's fatigue at the muscle level. Fatigue is generally caused by a number of factors, and some of the common factors are inadequate calcium. Well, that makes sense. The force of contraction is proportional to the amount of calcium nearby and the amount of cross bridges that are formed. If you don't have enough calcium, you can't create enough force. And this happens with repeated contractions. You can't release enough calcium. If you ever run out of things, and I thought about taking this out because it doesn't actually happen, but in theory, if you ever ran out of creatine phosphate, if you ever ran out of oxygen, if you ever ran out of nutrients, that would cause fatigue. Now, one exception to that is glycogen. the thing in the muscle that stores glucose, when it gets really low, and there's almost a level that you can predict, if you can measure it and you can see you get to this level, you can predict that a person will fatigue. Very tight correlation with fatigue and low glycogen. That's one exception to this depletion rule. A buildup of ADP and or an increase in acidity will cause fatigue. I won't go into the mechanics of it, but ADP is a signal in the muscle that turns things off. We'll put it that way. If there's a lot of ADP, that means a lot of ATP has been broken down, and it's a signal to rest. Here's the kicker. Acidity, the increase in proton concentration, comes from the breakdown of ATP. When you break a phosphate off, you also release a proton. Contracting the muscle using energy makes it more acidic. Not lactate. Breaking down ATP makes it more acidic. So take a second to appreciate, if you sprinted, did a lot of really high intensity work, you're using a lot of ATP, there is some spillover, and so those two things will happen at the same time. You'll see the muscle get acidic, you'll see lactate start to build up, and some people think that, oh, they're one and the same. Not the case. Lactate is the indicator that the muscle is acidic. And lastly, not even lastly, but you might not be able to transmit the recruitment signal. You might not get enough acetylcholine crossing the neuromuscular junction. You might not depolarize the muscle. And this is really interesting because this leads into a concept of fatigue being muscular or central. If you can't turn on the muscle, well, it's a deficiency at the muscle. But fatigue, as a concept, we don't really get it. We're still researching fatigue. Fatigue can be central. The brain might not activate the nerves. The nerves might not recruit the muscle. And this is one of the main mechanisms by which caffeine works. Caffeine makes exercise feel less painful, and it also helps to stimulate the muscles, to turn them on more quickly, even at low doses. It alleviates that central fatigue. Without it, or late into exercise, there are changes in the brain and in the way that signals are transmitted in the nerves that prevents them from turning the muscles on enough. So in short, a lot of things can make the muscles fatigue. And it's hard to pinpoint one, but there are some candidates, depending on the type of exercise and how long you've been exercising, that we can point our fingers to. Not the point of this class. The book takes a little tangent, diverts a little bit, to have you consider metabolism in recovery. Everything we talked about so far is metabolism to support exercise. All of those pathways make ATP that allow the muscle to contract. Well, after exercise is done, you don't just recover instantly. You don't catch your breath right away. You still have metabolism ongoing that's reflected by that heavy breathing, because you still need to make energy. You need to replenish the borrowed energy from the onset of exercise. Now, this says you keep breathing to pay back your oxygen debt. And oxygen debt is an antiquated term. It's not really used anymore, but it's meant to describe the fact that it takes a while to catch your breath. They call it oxygen debt. It's not reflective of what's actually going on. What's actually going on is that the cellular respiration is working to make ATP to restore phosphocreatine. Remember, we used this in a relaxed muscle. You're fully stocking the phosphate stores. Well, if they're not fully stocked after exercise, we need ATP from somewhere to do that. Comes from respiration. We've also probably used a lot of glucose in glycolysis. We need to build back up those glycogen stores. That energy, ATP, comes from respiration. We also need to reload oxygen. You need to pump blood at the heart. You need to restore ion balance. You need to release hormones and enzymes. All those things require energy. And in recovery, that energy is supplied aerobically. And it's reflected by you not being able to catch your breath. So at this point, you have a rudimentary understanding of how energy systems provide ATP for exercise and after. And if you're putting your books away already, you're going to be sad that you missed the 5 points on the summary slide, because they're not on Moodle. In this section, we covered a lot of information related to muscle, specifically skeletal muscle. Each fiber is a cell made up of many sarcomeres arranged end to end. And the sarcomeres have a number of different proteins, but those main filaments were myosin and actin, the thick and the thin filaments. Now those slide over top of each other in contraction to shorten the muscle, and the signal to do that is calcium. Calcium is released from the sarcoplasmic reticulum in response to the delivery of an action potential, and it specifically moves troponin out of the way and reveals the myosin binding site on actin, which allows the cross bridge to form and muscle to contract. We went over that in detail. The more calcium you have, the more cross bridges that form, the more force you get. But as the muscle shortens, that force, that tension changes. It's at its highest somewhere near the midpoint, and at its weakest at the extremes. And the reasons for that is when the sarcomere is too stretched at one extreme, there's very little or no overlap, no cross bridges, few cross bridges. On the other extreme, there's no space. Myosin is crammed into the Z disc, and it can't move any further. Highest in the middle, weakest at the extremities. That being said, the force that a muscle can generate depends on what fiber types are involved. Type 1 is weakest, type 2 is strongest. And we can increase force either by recruiting more motor units or stimulating the motor units that are already active. And lastly, the content of our lecture today, as we scramble to finish on time. Metabolism, the metabolic pathways we discussed, supply enough ATP to sustain contraction, to sustain the workload required by the exercise that you're doing. And those metabolic pathways are the creatine phosphate pathway, glycolysis, and cellular respiration.